and we will start this. Incidentally, if it goes longer than the time that we have allotted to this class and you need to go elsewhere, you should feel free to do that. I'm going to record it and I'm going to probably deliver it completely so that that background info is out there for you guys. You can watch it, the rest of it, whenever you, you know, have the time. So now I just get organized here. I'm going to share. Actually, I'm still going to share that, but what I need is this. And I've got to get rid of that. So much going on. I've got to get rid of that. I've lost my, okay, I can't see my mouse, but it's apparently there. We can see that? No. Okay, I got to get out of this. So annoying. Okay, desktop 2. Again, did I mention hiccups? Okay. All right, so this course is behavioral neurobiology. So obviously we should talk about what is behavior? What constitutes a behavior? And so there's a few pictures here. I like this one of the leopard seal and the penguin because it looks like the penguin is caught in like a vortex, you know, tractor beam going into the leopard seal's mouth. Poor thing. But anyway, there's a lot of different, you know, it's easy to think about some things as behaviors. Other things may not come to mind as readily as a behavior. So if somebody asked you, what is a behavior? What are some of the things that would come to your mind? And I think, here we go. For some reason, when I'm in this presentation mode, I can't see my mouse, which is not gonna work all that well. Let's see if I can. You guys should have an option to annotate. Do you see that on your control panel? Anybody see an annotate option? So if you click on that annotate and then there's a text option, I apparently can't do it while I'm in this presentation mode, so I can draw with my my pencil, but see if anybody can get something to type there. All right. Hello. A behavior is an action. Actions of animals. Okay, this seems to be a common theme of action. Based off of feelings or stimulus, eating, sleeping, conditioned response, a reoccurring pattern in the way an organism acts. So that's an interesting kind of adding some complexity there. Maybe something that animals do frequently. There's some people are selecting the same spot. Something externally observable, decision-making, interaction between organism and environment, sounds. Yeah, these are all really good thoughts. And an involuntary or voluntary response to a stimulus. So that's interesting. That adds another kind of level of complexity. Do you have to be able to control it for it to be a behavior or not? So let's see what some ethologists have come up with over time, and there have been a lot of them, to say that. So I don't know if any of you guys have had behavior courses before. Oh wait, I can't figure out how to clear the this is so funny. Okay, pretty much I have to do this. Okay, getting the hang of it slowly. So Nico Tinbergen is considered one of the fathers of ethology, the study of animal behavior. And he was really good at taking both kind of an observational and experimental approach to animal behavior. And a lot of really classic animal behavior studies were done by him. So his definition was the total movements made by an intact animal. But people have come up with lots of different ones. This second one here is about as simple as it gets. What an animal does is behavior. Others can be a lot more specific, like externally visible activity of an animal in which a coordinated pattern of sensory, motor, and associated neural activity responds to changing external or internal conditions. So that kind of definition kind of reveals a lot of the more complex things about behavior. You have the sensory world happening, and then the processing world, and then the response. And what elicits that behavior can be both a response to things that are happening externally in the environment and things that are happening internally in the animal, oftentimes in an interacting way. That's what hormones do, is change behavior by changing the internal environment in the mechanisms that control behavior. Lee Allen Dugatkin more recently came up with this definition. Behavior is the coordinated responses of whole living organisms to internal or external stimuli. And I think a couple of these have said kind of whole, intact, living organisms. And the distinction there is that, you know, if it gets hot and the body responds by say sweating, I think is an example Carew gives in the text. Is that a behavior? You know, so like what exactly constitutes a behavior? And so we'll just use that last definition for the purposes of our course, understanding that these definitions, you know, are variable and offer kind of different levels of detail. And none of them are incorrect, but people argue over this thing a lot. Scientists love to argue about semantics. Words matter. Okay, so if we think about the levels of organization, starting maybe at the smallest level of atoms and molecules, organelles, cells, organs, organ systems, individuals, populations, communities, ecosystems. What's cool to think about is they're all connected. How changes in molecules can ultimately drive changes in ecosystems. That's cool. And that's biology. Like, you know, everybody in biology studies some level or some point in this kind of level of organization. And behavior is largely concerned with at the individual level, but that doesn't mean that it's not important to understand the other levels, because behaviors contribute hugely to what happens to populations and ecosystems, and what happens below the level of the individual at the cells and the molecules is what drives the behavior. That's what controls the behavior. And so for behavioral neurobiology, we're basically going to be studying how these levels of organization generate behavior at the individual level. And you'll see that we're going to focus on molecules and organelles all the way up to whole organ systems. So Tinbergen, if you've taken an animal behavior course, you will have definitely focused on Nico Tinbergen's kind of organization of the field of ethology. And essentially he identified that there's 4 basic types of questions that you can ask about behavior. And those can be organized into a proximate level, which generally refers to like mechanisms, and an ultimate level, which generally refers to evolutionary mechanisms. So for proximate level, 1, how does the behavior develop? And then 2, what mechanisms activate that behavior? And then at the ultimate level, how does that behavior contribute to fitness? And then how did the behavior evolve in the first place? So where would you guys imagine that behavioral neurobiology fits in? We're getting a lot of chats, but I can't open them because I'm in presentation mode. I'm gonna guess you guys said proximate. And probably also in the mechanisms that activate it, maybe even in development. Again, that doesn't mean that behavioral neurobiology is not important to fitness or evolution. And in fact, lots of people study exactly that. How did neurobiological mechanisms evolve? And how do differences in the neurobiology of individuals contribute to increased or decreased fitness? So there's lots of different ways to approach these questions. And so these proximate questions of how does it develop, you get genetic developmental mechanisms like the effects of heredity on behavior, the effects of early experience or learning on behavior, the development of the sensory motor systems, and some of which are plastic and can change throughout adulthood and some of which cannot. So development is obviously pretty important. And this involves processes throughout the lifetime of the animal from embryo to adult. Then you can think kind of more immediately, what is the immediate mechanism that turns on or off a behavior? So you have these sensory motor mechanisms where you start with an environmental or an internal cue. So I guess the environment can be internal or external, right? The sensory system detects that cue. That's the first step. You have to have detection. And then you have the response system, which is presumably going to involve the nervous system. And then you have motor output, or an actual change in what the animal's doing. And so again, this involves processes that are occurring right around, immediate to the behavior that's displayed. And so by and large in this class, we'll be focusing on this proximate question. What mechanisms activate a behavior? It's really a problem that I can't see this chat because I don't know if you guys have questions. If you guys have questions, just unmute and be brave and say, hang on, I have a question. Because I can't figure out how to open up my chat, so I'm gonna figure that out. Okay, so the ultimate questions, how does it contribute to fitness? So essentially these are selective processes that shape the behavioral trait. So understanding better, how does this behavior change reproductive success or improve fitness or survival, both in the past and currently? So this obviously is focused on Darwinian selection, where you get a variation in the behavior, presumably because of the mechanisms that underlie it. If that variation is heritable, then you can get a difference in reproductive output of the individuals that show those different behaviors. And therefore you can get evolution that either enhances that behavior or represses it through a loss of the gene that controlled that. And just a note, as in all, most cases, the effect of the trait is on the individual's fitness, not necessarily on the group. Although we know that there are lots of altruistic behaviors that are pretty hard to explain and are really fun to think about and figure out how did that evolve if it hurts one individual but helps the group. The last question of kind of more historically, how did it evolve to come to the present state? And so this on the left there, kind of traditional cladogram, where you're mapping across time how things changed across time. So this is something that we're really used to thinking about for morphological traits, but maybe not as used to thinking about behavioral traits, partly because it's a little harder to get at those questions. You can look at relationships amongst living organisms, and if you know their phylogenetic history and how they relate to each other, then you can start to understand some of these things. And this paper in 2018 was basically investigating the acoustic and the display diversity in birds of paradise, which are fantastic. If you haven't ever explored birds of paradise, they just have such an amazing array of courtship displays that have become really sexually selected in really extreme ways that are pretty cool. But this is a really beautiful cladogram in how they've kind of arranged it, and demonstrates the huge diversity that's evolved in these systems, these birds that are relatively closely related to each other. Okay, so we need to talk about Tinbergen's zeroth question, which is not in those 4 previous categories, and that is just observation, which is the starting point for science, is observing something that makes you ask why or how or what. Tinbergen was really good, you know, the history of science is actually really fascinating, and how it kind of changed over time. And we're in a current moment of kind of extreme focus on experimentation and deduction, but it hasn't always been that way. And there was a lot of just kind of watching and speculating without making experiments before, certainly before the 1900s. And then throughout the 1900s, we've seen a shift to designing controlled experiments to try to test ideas. But they start with ideas. So Tinbergen was really good at watching animals and asking what seemed like really simple questions, but that ended up revealing a huge amount and kind of starting the study of behavior, the ethology. People hadn't considered it really a science before that. So he got his ideas by watching animals. And so I'm going to show you this video. This is one of his classic studies of a gray lag goose. I think I spelled that wrong. I think it's EY. But anyway, where these geese, what he noticed is that they have a really strong instinct to return lost eggs to the nest. But what was fascinating, let me see if I can get this to go. Yeah. Is what happens if you pull the egg away in the middle of the roll. So here's a foal. She's going to get it all the way into her nest on that one. And now he's going to pull it out. And basically what he noticed is that the goose will continue the action all the way to completion, regardless of if you take away the egg. And with some more kind of experimentation and thinking about this, he coined the term of, there we go, a fixed action pattern. So a fixed action pattern is a behavior that I think a few of you in your kind of what is behavior in that in your messages kind of were getting at, this idea of kind of repetition and stereotypical. A fixed action pattern, not all behaviors are fixed action patterns. They're very specific behaviors, but here's the criteria for them. One is that they're innate, so they don't need to be learned. They're instinctual, they're encoded in the genes, and you don't need to learn from a parent how to do it. The second is that they're very stereotypical. So the same motor pattern is, when it's triggered, is executed in the same way each time. So it's really stereotyped. And then it doesn't require sensory feedback once it's initiated. So some sensory info has to trigger it, but once it starts, you don't need it to keep it going. And that's pretty cool. So we say that a fixed action pattern, the behavior, is released by a stimulus in the environment, and that stimulus is called a sign stimulus. And basically what it does is some external cue, this sign stimulus, triggers a neural process that turns on the fixed action pattern. And that whole process is called the innate releasing mechanism. And so it's basically just the sensory neural motor pathway that controls the behavior. And that's what we're going to be looking at a lot in this class. We'll look at fixed action patterns, we'll look at reflexes, and we'll look at more complex behaviors too. So releasers can also vary in their specificity and their potency. So in other words, they're releasing value to triggering a fixed action pattern. And some of them can be released by a pretty broad array of cues. So in other words, there's not a lot of specificity. So the gray lag goose again, I'm gonna actually, oh wait, I can't. Oh, hey, there's my mouse, it's back. I don't understand where it was. Okay, so there's some nice music going along with this. It's not gonna let me fast forward it. We get to watch it all. So there's a cue ball, which looks a little bit like an egg, right? It's round, parts of it are white. But it turns out that that's not necessarily, it's not necessary,
An egg,
as you'll see.
Needn't be white, or even egg-shaped.
Light bulbs.
In her eyes, even a wooden cube can pass for an egg.
Okay, some nice background music to that video. Anyway, you can have, you know, some behaviors are so strong and they're so, you know, this kind of a fixed action pattern can be triggered quite easily. Other things require some more complexity. So some releasers are more complex, requiring movement or a particular position of the cue on in the world, or a particular context for the fixed action pattern to actually be initiated. So in this example, this is again Tinbergen. He created this shadow, this shape, I guess, that can either, if you move it in one direction or the other, can either look like a goose if it's flying to the left, or it can look like a raptor if it's flying to the right. So this takes advantage of the fact that a lot of animals have kind of group specific attributes, and that prey species like songbirds, which is what this experiment was done on, pay attention to those attributes, and that those might be what matters the most in their response. And so if you move this shadow, if you just keep it in one spot, they don't necessarily respond to it. If you move it to the left and it looks like a goose flying, they don't respond at all. But if you move it to the right and it looks like a raptor flying, then they alarm call and they seek shelter. So it's not just the shape, right? In this case, it's the context of how it's moving, how that shape is moving, that triggers the fixed action pattern. So there's something about the sensory processing of a shape like this moving in the right direction that turns on the alarm behaviors and the anti-predator behaviors. Here's another Tinbergen example where three-spine sticklebacks are really famous for being super aggressive. They're actually used in labs a lot of times for behavior classes because they're easy to keep. And this is a male. He's got a red belly and he's very aggressive to intruding males. So if you give if you put any one of these stimuli into his tank, he won't attack the actual stickleback fish if it doesn't have any red on it. So if it's female or if it's a male that's not displaying red, he won't attack it. But if you put any one of these rocks of various shapes that have a red underside painted on them, then he'll attack those. Similarly, if you put, or again, position specific, if you put the red on the top, he no longer attacks those stimuli. So again, you know, some releasers are really broad and generalized, and some are much more specific, pertaining to, you know, movement or positioning. So here's another example where an animal is using cues in the environment in order to navigate. So this is a experiment with bee wolves, and so otherwise known as digger wasp here, carrying a paralyzed bee back to the nest. And what Tinbergen did in this experiment was put a bunch of pine cones around the opening of the nest. And every time that that bee wolf leaves, they do a circle of the area and then they leave. And so that's what he noticed. And he was curious about that. And so he put these cones out to see if maybe what she was doing was taking note of landmarks so that she could find her nest again. And what he did then is moved the pine cones. And when she came back next, she went to the pine cone location, not to the nest location. This is probably what we would do too. If somebody moved all the houses we normally take note of when we're finding our own, we probably wouldn't be able to find our own house for a little while. So it's not that this is a simplistic model, it's just how brains work. Incidentally, if you change the landmark to some other kind of thing, she'll still go to the correct spatial configuration, not necessarily not the type of the landmark. So she didn't then go to the triangle of cones, which did mark the nest. She went to the circle of rocks that resembled the circle of pine cones originally. So anyway, all just to show that there's kind of complexity of some of these things. Another interesting thing is when these fixed action patterns can happen in sequence between two individuals, releasing fixed action patterns back at each other. And this is really common in courtship displays. So here's a video of, oops, I guess I paused it, of some of these birds of paradise. This is the male who's acoustically displaying, and now he's going to behaviorally display to attract a female who comes in. Birds of paradise are really famous for being really ostentatious in their displays. So there's the female, and so what he's done, he just triggered a fixed action pattern in her. And when she did that, she's kind of reinforcing and triggering fixed action patterns in him. He's making those noises with his feathers on purpose, which is pretty cool. Okay, so you can get, interactions that are really complex as well. Okay, and then finally, a very brief overview of learning, which is going to feature centrally in this course too. And associative learning is basically when there's an association between releasers and these environmental cues and behaviors. And those can be strengthened or weakened through either positive or negative consequences and pairing those consequences with the behaviors. So a really classic, it's even named classical conditioning, classic experiment that Pavlov did was with dogs. And basically what he did is he knew that food, which is an unconditioned stimulus, the dog knows what food is, and that the olfactory smells cause him to salivate. Those are the unconditioned stimulus and the unconditioned response. Then what Pavlov did is every time, he paired a tuning fork, which normally is a neutral stimulus, doesn't cause a response in the dog. It's certainly not a salivation response. But every time there was food, he paired it with pinging a tuning fork. And so eventually, through neural associative learning mechanisms, the dog learned that a tuning fork sound means food is coming. And so eventually you could take away the food, just ping the fork, and you would get the salivation response. So that's now a conditioned response to a conditioned stimulus. So that's a classic form of learning that we will go into when we learn more about this animal, Aplysia. This is their sea slugs that turns out to be a really good model for understanding the neural basis of associative learning. So we're going to learn this for now. We're just going to look at it as that it exists and that there are, we understand at least some mechanisms underlying how neurons change their synaptic activity and their connectivity in order to pair a stimulus with a learned response. And that you can actually map this in some organisms, like in the sea slug. This is a reflex, but you can associatively learn to do the reflex in different ways. And then we have operant conditioning, which is essentially where you're just conditioning and rewarding a particular behavior. So like a rat pushing a button to get food, the food is the reward. And so they'll quickly learn to push that button over and over and over again to get a lot of food. If instead you give a shock, then they very quickly learn not to do that behavior. That's called operant conditioning. And also has a neural basis. And again, there's some examples in aplesia that we'll get into. Another key attribute of learning is that memory is required for associative learning. And there's two general kinds of memory. There's working memory, which is a really short-term retention of information that allows you to complete tasks that are otherwise unimportant to remember. So like when you cross the street and you look left, you have to be able to remember if there's a car coming or not as you look to the right, or else you're caught in a perpetual looking, right? You can never cross the street because you can't remember what was happening to the left, even though it is completely unimportant to remember what happened to the left 5 minutes from now. It's very important in the immediate term. And so that's working memory. It's memories that you need to do a task right now that you're not going to bother to store. You don't want to waste neurons, remembering that there was a car coming down the road on the 15th of July at 2:56. It doesn't matter unless something else happened that makes you remember, like that car hit your friend. Now you're going to remember that whole scene. But normally for working memory, it's information that's not going to be retained. Reference memory is essentially long-term memory, and it's required in order to learn general rules of a task. So an example I gave here is figuring out a winning video game sequence, right? You remember that you had to, jump on this square and, whatever, do a particular order of tasks in order to reach a goal. And that we do, all the time in life as well. And just like for associative learning, we're starting to identify the underlying neurobiological mechanisms for that. So this is an example in fruit flies where a mechanism has now been worked out involving dopamine for making an association between an odor and a shock and getting that fly to stop doing the behavior. And then how to reset it using dopamine and these kind of neurobiological mechanisms. Okay, so that's that introduction. And basically, we're obviously a little bit over time here for class. I'm gonna stop the recording somewhere. And we will, I'm gonna be posting, actually I'll keep that recording going in case somebody needs to watch this later. So right now on Canvas, there are 3 other videos. This one will be posted there soon too. And the way that I'm doing videos is I'm actually uploading them to a YouTube channel, and then you will access them on the YouTube channel. That's the easiest way to get them there for a long time. Because Canvas and whatnot has limitations on how long you can store videos. So they'll be there for the entire quarter, and you can access them whenever you want to or need to. And I'll be posting a worksheet that isn't going to be due. This worksheet will just be kind of guidance for, can you answer questions about this background info? And hopefully will help you to feel comfortable going into the quiz next week. I'll also then be posting videos on our next unit. So this week is more intensive than most because of the background info. But I'll be Thursday and Friday, by the end of the day Friday, there will be the videos posted for our first unit, which is echolocation in bats. And so you should watch that enough to be able to answer those topical, did you watch the videos and prepare for class questions on the quiz next week. Any questions about that? I'll probably send an email as well. Go ahead, Jennifer.
Will you also post the lecture slides as like a PDF or a PowerPoint for the lectures?
Yes.
Okay.
Yes. So those are already posted for the videos that are already up there recorded. I'll post them today's, this behavior lecture too, so that you guys have those. Okay, well, if there's no other questions, then I'll be in touch by email soon and I'll update the schedule and whatnot. And I will see you guys on Thursday. Okay. Bye, everybody.